In this video I will go through how you get going with OmicLoupe, and I will show you how we can load the example data that is provided together with OmicLoupe and how to get that one into the system. First, this is the GitHub page, and here we have instructions on how we can do the installation. So it's an R package and you can install it directly from GitHub using this single command, using the DevTools package. And you might need to install some dependencies. You will see if the installation fails. For me, running on Ubuntu, I needed to install the 3 packages. If you run on some other system and you run into other problems, feel free to send me an email and I will try to help you out. And so the example data is present if you go to the releases. So together with the release, I have provided a dataset. So you have the source code, but you also have the zip file here. And I will download this one. And then I will open the downloads folder and I will extract it here. And it will look a little bit different on Windows, but the logic should be the same. And in this folder now, we have 3 different TC file, tab separated matrices, and we have a README. And the README outlines where this data is coming from. So just a brief moment show you it's coming from this study, and they have done a great job comparing different proteomic datasets where they have the same dataset and they have processed it using different software methods and comparing the results. So I have processed the data provided by them, into a format which is compatible with OmicLoupe. And so I outlined this in a previous video, but we will take a quick look at what we have inside this one. Now LibreOffice didn't open, so I might need to... Okay, we open it as a text file here. So we see here in the design we have the sample names and we have the condition. So it's a tab separated by tab, and one of the columns should match the dataset and one of the columns should match can contain other conditions related with the sample. So if I open here how it looks, so we see the column names at the top here. So you have first some annotation columns. Let's see if we can open it in a spreadsheet program instead. Yes, now it worked. First we have annotation columns, so general information about the proteins. Then we have the statistical columns. So we have 4 columns here which are related to a statistical comparison we did between known levels of different proteins. So in this dataset is a technical dataset, so they know what's supposed to change here. But so for each of these comparisons, we have the average level of the protein. We have the log fold change, so this is the difference in a log 2 scale between the groups. And we also have the p-value and adjusted p-value for each comparison. And the p-value you generate for each statistical test, and then the adjusted p-value, then you correct for doing many tests by calculating this for the whole set of p-values. And then finally we have the sample columns here. And if you have data in this format and you don't know how to generate these statistical columns, so you can do this with many R packages, such as the Limma package or the DSEC2 package if you're working with RNA-seq data. If you're working with proteomics or metabolomics or some other normally distributed, so it's proteomics, metabolomics or microarray, then you can use a software called the normalized DE. So if you go to computational proteomics here, this is another software. I will not talk about it in this video, but here this is another R package you can use to perform normalization of your data and also to calculate statistics, and then you will get it in a compatible format. So we can go to this web link also, and then we can run it directly in the browser. So that's one way to do it. So then we know we have the annotation columns, which is the first ones up to the PEP count, roll-up score. Then we have the 4 statistical columns, and then we have the data columns. So with that, we're ready to run OmicLoupe. So I will open R, and then I will use the OmicLoupe, and then run app is the name of the function. So the first part is the package, and the second part is the function name. And I will run, and then it will open in a browser. And if it doesn't open, you can just copy the link you get to your browser. And so here we need the dataset and we need a design matrix. And you can do this with one or with two datasets. So if you start with one, I click browse, and you can go to downloads, and we'll take one of the datasets. So now I selected one process with a software called DIA Umpire. And we can select the second one, and we get the design. And we can see the columns, the column names that is present in the data matrix, and here in the design. And in the design there is only two present in this case. And and we can run this command identify columns, and if you run this, it will look for the column in the design matrix which matches the column names in the data matrix. And in this case it found these 6 names. And then it will also look for these kind of statistical columns I talked about, where they follow this pattern. So in this case the comparison is called comp, and then we have these 4 different ending parts. And if you have similar columns which are formatted in a different way, you can go to the table setup here, and then you can change the setting for what's the different endings. So with that we're ready to load, and now it loaded, and it found a little bit more than 3,000 entries. So if you only have one dataset, this is enough to get going and start exploring. So now I will load a second dataset. So then I will click this box here, 2 datasets. And if we have different datasets, not coming from the same source or different samples, then we will load another data file and another design file here. In this case, it's the same set of samples, just processed in a different way. So then we can use this setting here, match samples, and then we only have one design. This can be useful also if you have, for example, transcriptomics and proteomics from the same set of samples. And we have additional settings here. I will talk about them in a moment. So we will load the other one. Either you browse or you can just drag directly in, and then it loads. And it shows the columns. We need to run the identify columns again because these are not found yet. And so now it has identified the columns. And one thing is important here is that we need a matching column between these two. So in this case, because they are the same, they have the same annotation columns, it's totally fine we can use these protein IDs here. If we would come, for example, our RNA-seq data here, we would be more careful with that the columns actually matching. And with that we can load the data. And it's also, part of this is calculate correlations between all the features. If you take too long time, you can check the box here, skip correlation, and it will go quicker. And if you have multiple duplicates of protein IDs or the type of IDs that you're using, then you can either pre-process your data to make sure that there only are unique IDs present, or you click this one, discard the dupes here. But now the data is loaded and we're ready to start explore the data, and I will show you that in another video. So with that, if you have questions and problems, feel free to reach out to me. And